everyone. It's weird not to see all of your faces, but anyway, we are starting to get used to it, I guess.
The title is a little bit different from the one that was announced on the program, and main reason is because I'm going to focus. Hervé told me that I have 1 hour and 15, and although the title mentioned artificial life and artificial intelligence, I've decided to focus essentially on artificial life, which I think is much more related and much more connected with astrobiology, and because you know, like you guys coming from perhaps physics, astronomy, or biology, there are a bunch of computer scientists who are interested in trying to understand life, but by means of software, computers, robots, so artificial means. But this is, you know, artificial intelligence, something different. You know, the people there are more interested in trying to understand cognitive processes and help cognitive processes, while artificial life is much more related with theoretical biology, so deep biology. So this is the reason why I've decided to focus only on that. Otherwise, that would have taken me much too long to cover both fields. So the title now is The Origins of Artificial Life and the Artificial Origins of Life. So you see there's a word game. And I'd like first, you know, before really entering in my talk, I'd like to apologize to some extent, because I thought I was very happy and you know, and happy to contribute to this school. Is not the first time. I must recognize that I'm not so much involved with this field any longer. I'm coming back to artificial intelligence much more. But yet, I hope that what I'm going to tell you, you'll see that most of my reference, the things I've myself written, are a little bit dated, something like, you know, 10 years old or sometimes 15 years old. But I'm still very much interested in this field, even if myself, I'm not really working in that any longer. I organized the first A-Life conference in Europe. That was done in '91 in Paris. Then I organized the second one in Brussels. So I've always been interesting to try to understand and following up what was happening in this artificial life field. But myself as a researcher, I'm not so much dedicated to that any longer. But yet I think I can testify about what it is and what use you could do from that field. So I split it in 2 parts. First one will be to cover a little bit this topic of artificial life. And then I know you're supposed to be interested in the origin of life. And I'll show you in the second part, so there will be 2 parts. I'll show you in the second part what, according to me, the researchers in artificial life, you know, has a scenario in their mind, you know, the scenario for the origin of life. So you'll see that, you know, a lot of artificial life, you know, researchers, former and still, you know, modern researchers in artificial life are still trying to understand how life could pop up, how life could emerge. And you see that they have a certain kind of scenario, a temporal scenario, a temporal chronology. And I'll try to show you the different steps that they have imagined for the origin of life. So first artificial life, and then the origin of life. So, well, you know, there's a long tradition of, you know, using computer software and for robot for trying to understand theoretical biology. So there is a kind of natural fit between theoretical biology and computer science. And basically, artificial life, is, if I had to summarize that, I would say that it is theoretical biology done by computer simulations and really nothing else. People are trying to understand the rule-based qualitative mechanism and function of life. You know, if you're a biologist, you would sometimes given too much importance to the component and the materialization of life, and sometimes you neglect or you give less importance to the functional aspect of life. As you know, computer scientists in general, they adopt a kind of philosophy which is in general called the functionalist philosophy, which is the idea that the material is not so much important. For computers to run, you could substitute a mother card or a motherboard with another one, and it will run exactly in the same way. So the computers, don't care about the materials inside the computers. So what a computer is doing is basically, realizing some functions. And so we're not so much interested in the, materialization of life, but we're trying to understand with other means, the silicon means, the material that are being constitutive of our computers. We try to understand the functionality of life. We're not trying to define, life researchers are not trying to define really life. I think this should result from a kind of consensus, and the researcher in life doesn't want to take a strong position, but they are really interested in trying to simulate or understand better a lot of mechanisms that, most of the biologists will generally accept to associate with life, like those ideas such as self-maintenance, self-organization, things like metabolism, autonomy, self-replication, evolution, all those famous, topics that are all related with this notion of life. Computer scientists are trying to make them clear, to explicit them through software development. Why software models? Well, they've been always around. Those software models were very used in engineering, they're very used in natural science, they're also used in theoretical computer science. And in fact, I'm in the middle box, I'm just referring to some of the artificial things that are very popular, like artificial life, artificial intelligence, artificial reality, but I've myself even created a community which was named Artificial Immune System, where we try to take inspiration from immunology in order to build interesting engineering artifact. And what is the use, the main, the main exploitation of those computer simulation? I think they can play different role. They can have, different, motivations. One of them, which is a very, simple one, is for teaching. I guess computers and software are a fantastic media in order to teach some basic ideas. And I'm just referring here to Richard Dawkins, who you certainly know, certainly one of the most important scientists of this of the previous century, but also the current one. The author of The Selfish Gene and The Blind Watchmaker and a lot of other very famous books. Definitely one of the most famous evolutionary biologists. And I remember Richard Dawkins in one of the first A-Life conference, he was invited a long time ago, and he showed us a model called the biomorphs. And what he was trying to convey by showing the simulation that you can easily see here, what he was trying to convey is that simple evolutionary mechanism, like in computer science, we have an algorithms called genetic algorithms, who is trying to take any kind of codes and it recombine codes, so a kind of genetic crossover. It mutates codes, so in order to create new code, and the best code are being selected. So very simple evolutionary mechanism. Richard Dawkins was showing that, with a simple succession of a genetic mechanism, you could generate very soon and very easily very complex shapes. So some kind of a insect-like shapes that Richard Dawkins called biomorphs. And that was his answer to some of the, not scientists, people defending, for instance, the creationist idea. And some of the argument that those people invoked is this notion that, it's hard to get something as complex as an eye or as any kind of, organic function by just the, evolutionary mechanism. And Richard Dawkins was showing that we don't understand the evolutionary mechanism. And by replacing them in a computers, he could show that in very few generations, very few iterations, you can reach an incredible complexity by just these simple ideas of mixing things, mutating things, and selecting the most adapted or the most fitted things. Another thing that I've played around a lot was the simulation of the immune system, and where you could easily understand, the first answer, the first response of the immune, and the second one that you can see here are much faster and much powerful than the first one. And just to explain to immunology student who is running the immune system. So for instance, why do you vaccinate people in order to make a second reaction to a virus attack much more effective than the first one? And that is for teaching, but you can also use, obviously, we're scientists, so we like to build model, And we build model for doing prediction, in order to, confirm the model or disconfirm it. You need to be able to make prediction. But those predictions can have different level of precision, and it's still very interesting sometimes to make some kind of qualitative prediction. And I'll show you some demos, some software able to create some form of self-maintenance or able to self-reproduce. And even if it's not exactly fitted to the reality of the biological system yet, I think that if you're able to reproduce in your software some even very qualitative mechanism like, maintaining a cell, or self-reproducing any kind of organism or whatever. And if you understand the principle that underlie those, processes, then I still think that you are contributing a lot to our scientific understanding, even if the mapping between the reality and your software is still very qualitative. We're not mapping numbers. We're not at the level of physics where we try to map numbers with the, the fifth decimal. We're just trying to map a qualitative phenomena with a qualitative phenomena produced by a software. But yet, if it matches, we still have some good lessons to be drawn out of the software mechanism. And obviously, the ideal would be to reach a level where we can really do what we used to do with mathematical modeling, to reach the level, the quantitative level, where our model, is able to produce some numbers, and those numbers can match the one that we have in our computers. But this is often the dream of science. It has to be, because numbers are much easier to compare than just qualitative phenomena. I can tell you that in artificial life, very seldom, we haven't reached that level yet. Most of the models are very qualitative. They help us to understand. They help us to have some ideas of what's happening in the real system. But we don't have the final confirmation, which is the one that normally numbers provide. If the numbers map, so this is good. We have not reached the quantitative level yet. We're really still stuck on the qualitative level, but still very useful. It's not because it's qualitative that it's not necessary to have them. And in most of those models, because we're qualitative models and because we're computer simulations, big problem is when I'm showing the model to most of the biologists, whatever the immune model or some models that I'm doing in neural networks with the brain, or some other genetic model that you can do, most of the biologists will tell you that this is much too simple, this is much too abstract. But yet, even if they criticize the model for this level of abstraction, I believe that it's not because, any kind of model has to abstract somewhere. So the whole issue is what level of abstraction is useful, as Einstein even said many years ago. The model can be even very simple if it's still useful, if it still contribute to your understanding of the reality what you are interested in, if it makes you progress in your understanding of this reality, it's still valuable. You don't have to worry if your abstraction is too abstract to some extent. And I'm showing these two paintings of the same city, the Italian Venice, that was painted by Canaletto, and then just 50 years later, or perhaps 100 years later, by another painter called Turner. And they both show Venice. And obviously this one is really showing Venice in a kind of very qualitative way. You can still guess that this is Venice where, while Canaletto was very precise in his painting. He was so precise that it was even possible to exploit his painting to understand how high the water reach, which level they could reach during big, dropping and big, even the water, even in Venice, the acqua alte, like we call it in Italian in Venice. Another, obvious things between biology and computer science is that they really look alike. In fact, this is the reason why they both influence themselves for a long time. I guess biology, and I'll clarify that later, I will say that biology and computer science and informatics, they do function at many levels of abstraction. Computers, and I'll show you that in the next slide, computers is just a set of abstraction, and you're not playing with the transistor of your computer. You're playing with, for instance, what perhaps some of you are programmers. When you program, at the end, there are some transistors that let the current flow, or transistors that are blocked, or transistors that are open. you don't see that because you program in Java, whatever, in Python. But yet, underlying that, those things, you have a lot of transistors just opening and closing themselves. And that is, each level is functionally neutral while embodied in below a level. You're not playing with the transistor, but what you do in Python or Java is at the end, playing with the transistor. Most of the computer scientists will, accept that at the ultimate level. So, for instance, what a transistor is doing, but mechanisms have to remain simple. So we are in quest of a kind of underlying simplicity. We get the feeling that this is the Occam razor type of thing, that underlying everything, it shouldn't be too complex. The lowest level should be simple. And in computer science, the lowest level is very simple. A transistor is just a kind of interrupter, making a 0 becoming a 1 and a 1 becoming a 0. And in fact, you could make all your computers just as a bunch, an assembly of very simple, what is usually called gates, but very simple Boolean gates, things that even a 5-year-old kid could understand. But obviously, you combine those things in very different way, in very complex ways. But the elementary things are still very simple. Another thing that computers can do that is, connected with biology, is that they use brute force. This idea of brute force is very important in computer science. Computers is a dumb, computer is stupid, but by just doing a lot of times the same stupid things, so iterating so many times, then at the end it can produce a chess player or a Go player. It can drive the car, but it can do that because it has tried so many things, and there was a lot of garbage in what it tried. So there's this idea that perhaps biology, like computers, can generate many, many, many trial and errors, and they exploit brute force for making emerging solutions to very complex problems. So for the difference level of abstraction, let me just, you don't need to be computer scientists, but you could imagine that the transistors would act like molecules. And you combine the molecules, here the transistor, in order to create gates. Or you could say molecules can create genes. Here, this is gates, which are a combination of different transistors. And then you combine those gates to do circuits. Here, for computer scientists, it's a famous circuit called an adder. It's something that allows you to add binary number. And then you see that, here you have an addition, but nowhere here you have an addition. Nowhere here you have an addition. But when you combine those gates in a certain way, you can do the simple binary addition. 10 plus 11 give you 101, which is a binary addition. And this is how computers work. You put together, you assemble things so that you realize high level function that you are interested in. And you know that even when you're not doing the addition this way, you just write in a computer code A plus B, but at the end, the A plus B is some current that flow through those different gates. The big difference between biology and a computer scientist, biology and engineering, and I'll come back on that later, is that in biology, even if you have higher level, of functionality that emerges, and that was the title of Rich Dawkins' book. In biology, the engineer needs to be blind, in the sense that in computer science, initially what kind of functionality you want to achieve, and then you will assemble the block in order to achieve that functionality. So it's a top-down processing. You have the top, you want the top, and then you try to assemble the blocks in order to achieve the top. That can be the case in biology. There is no engineers in biology. So how does an interesting top, an interesting level emerges? This is one of the big mystery of biology, and I show you how the A-Life researchers try to answer this mystery. Well, the connection between computer science and biology has been done many, many times. In fact, a lot of researchers in different fields, even in cognitive science, but certainly in theoretical biology, used a lot of computers analogy. And here, this is a book. I think the book is a bit dated now, but it was a very nice book. I remember reading it, I guess, 10, 20 years ago. which the book was A Computer in Every Living Cell. And in fact, it was trying to understand cell by means of those simple, computer process. Here you see a transistor, and the title of the chapter was Protein Switches. And these are physicists and computer scientists. None of them are the label of biologist, but all those famous guys, had an important impact in biology. This is, another proof that theoretical computer science, theoretical physicists can have an important contribution to theoretical biology. And some of those people, you might recognize those faces. This is Erwin Schrödinger with this famous, piece of, masterpiece entitled What's Life? This is Alan Turing, and this is John von Neumann. This is the both granddad of computer sciences. And interestingly enough, those both guys, they've been also the fathers or the grandfathers of artificial life. Alan Turing did a lot of interesting works for understanding morphological process that have become very famous even in theoretical biology. And John von Neumann has done so many things in theoretical biology. He was one of the pioneers of neural networks, which was a lot inspired from the brain. He was also one of the pioneers in understanding self-replication, and I will come on that later on. This is one of Belgium one who's working at my university, passed away a lot of years ago. Name is Ilya Prigogine, and it was a Nobel Prize in physics, but he wrote so many things on the origin of life, on theoretical biology. One of his major ideas was this idea of a bifurcation and dissipative system, how the system can self-organize. So this idea, a lot of those ideas related with self-organization comes from him. This is Freeman Dyson, who is an astronomer, physicist, but also, has written a lot of things on the origin of life. This was a workshop that has been organized a lot of years ago with some theoretical biologists, but also some strong physicists. If my slide is precise enough, here you can see Stephen Jay Gould, Lewis Wolper, and John Maynard Smith, Stuart Kaufman. They didn't present themselves as theoretical biologists. Some would, some not, like Stuart Kaufman, who is a real physicist, but yet, they were exchanging a lot in trying to understand the fundamentals of biology. An interesting comparison here between a very famous, French inventor back in the 18th century during the French Revolution called Vaucanson, who created the first, artificial organism like a duck. This duck was a first kind of artificial automata able to some extent imitate some living organism. And this is another physicist, important figure, who is the inventor of artificial life, called Richard Langton. And he made a proposal in a famous paper in my community entitled Life at the Edge of Chaos. So he wanted to understand life, and he believed that you could use cellular automata for understanding living process, like for instance the game of life that you may be familiar with. And he was trying to show that life was poised at the edge of chaos. So life had to do with not chaos, not regularity, not fixed point, not oscillation, but some kind of intermediary process, which take granted from the richness of chaos, but needs some form of stability too. And this is another one called Doyne Farmer, who again wrote a lot of interesting papers on the origin of life. What biology can expect from life, as I was telling you, pedagogy, but also a lot of study on key qualitative principles underlying life. So like the Turing morphogenesis, the von Neumann self-replication, things like the scale-free networks, attractors in complex systems. This is all ideas coming from physics and coming from computer science that should definitely inspire, and that often are ideas that are inspiring biologists, when they do their daily work. So for instance, John von Neumann, who is, according to me, the most incredible scientist of the 20th century, he was living in he was working in Princeton, so he has his office close to Einstein. But I guess that even if, people often mention Einstein as being being, the greatest mind of the 20th century. I guess when you see the achievement of John van Neumann, the creator of computers, but also the creator of quantum mechanics algebra, which is very famous in quantum mechanics, the creator of neural networks and so many other ideas. And he invented also the concept of self-replication. And it's interesting to know that John van Neumann, we were back in the 50s, and no one already got the Nobel Prize for discovering the DNA, which, as you know, is the first really well-known self-replicator. And we were back in the 50s, just after the Second World War. But John van Neumann understood that, there was some kind of replication in cell, people knew that, but he wanted to understand deeply the principle of self-replication. So he made it kind of mathematically. He believed that in order to have a self-replicator, you need to have a certain constructor, what he usually called the universal constructor. This is the C here. And if you want to construct a machine, for instance, this machine according to a certain plan P, well, if you have this universal constructor, the constructor will receive a plan of a machine. P is the plan of the machine. And then it will construct the machine according to this plan. So you first need to begin with this universal constructor, able to construct any type of machine here according to the plan of a machine. So you receive a plan and you construct the machine. But then the question that came to the mind of John van Neumann was a simple one: how the constructor can build itself. So how the constructor can self-replicate, create another constructor exactly equal to itself. And then he thought about it, and he said, okay, in order to do so, we need not only the constructor, build the machine according to the plan, but once it has built the machine according to the plan, it needs to, assemble the plan to the machine. So to glue, to some extent, the plan to the machine. Because if you have a constructor able to do so, then you see that if a constructor tries to construct itself according to the plan, so naturally you have these things going on. So CPPC gives you CPPC. So you have a full self-replication. You can see that. And it's interesting to see that even in natural cell, you have some way a constructor and a plan. That was invented much before the DNA discovery, the fact that the DNA is a form of plan for the protein, which are able to construct the cell. But those abstraction, can be very rich, very interesting. And in fact, van Neumann built also the first cellular automata, which was able to self-replicate following those same ideas. So the computer scientists, the software engineers can give a big hand to theoretical biologists, because I guess it can help him to understand things like, when biologists say life is an emergent phenomenon, for instance, what do they intend by this notion of emergence, which is a very used notion in computer science and biology. So can we understand better what is this notion of emergence? In fact, one of the book of Luigi Luisi, who's a famous theoretical biologist in Italy, trying to work on the origin of life, was life as an emergent process. But what is this notion of emergence? What this notion cover? We know also a lot of biologists would tell that life owes a lot to randomness. When you're a computer scientist, randomness does not exist. And because computers is by definition fully deterministic. So you don't have any strong randomness in computers. So what kind of randomness are biologists referring to when they are interested in random processes? Is this deep randomness? Is this the same kind of randomness that you find in quantum mechanics? Or is it just the fact that we don't know, a kind of unknown randomness, like most of the random process in probability theory? You'll see that I'll mention autocatalysis soon enough, because this is another thing that interested me a lot. Life is a self-regulated process. Computer help to understand what is this notion of self-regulation. Langton, the kind of Indian guys that I showed you before in one of my previous slides, was thinking that life was appearing at the edge of chaos. And why so? Because the edge of chaos was a dynamics that was able to process information. So it is the first type of dynamics that is able to some extent to replace and to behave like a Turing machine. So according to Langton, life could only start when you had this capacity of information processing, like, for instance, a Turing machine can. And so software can really help to disambiguate what a lot of theoretical biologists would have in mind when they try to understand the minimal life processes, the basic life processes. And this is something we share with exobiologists, because I know that a lot of exobiologists are trying to understand or trying to discover or trying to reflect and think on, what could be another form of life, or what could be another kind of life on another planet, And so this is kind of sharing ground, because computer scientists, they're also interested in trying to understand life with different materials, life with different kind of, emergence, and that could be a connection with exobiologists too. So, for instance, emergence is a very interesting and key concept in computer science, because computers help to understand interesting collective phenomena, which, when you see those collective phenomena, you're being surprised, because it has nothing to do with the process that generate them. So with computers, you can do that easily. You code, for instance, the behavior of some elementary cell, and you code elementary local rules making those cell interactings, and then you see collective phenomena which might surprise you. And when you're surprised, this is often the starting of an interesting process or interesting discovery. In my lab, we're doing a lot of research on emergence computation. So, for instance, if you go on YouTube, you'll see that we have been a lot inspired by biology to build what we call swarm robots. This is robots that are very simple, but when they do connect, this has been inspired by insect and other kind of very simple animals. When they do connect, when they do create swarm or assembly, they can achieve tasks that none of them would be able to achieve alone. It needs to connect. So, for instance, this here, you have here a small group of 3 robots. Those 3 robots might be able to go over a gap that they wouldn't be able to go over if they were just alone, because any robot will fall into the gap. But when they connect, they form a shape, and this shape helps them to go over the gap. And we are a lot interested in ants and society of insects in general. And you know that ants can collectively find shortest path when you have an alternative. The ants, by some form of emergent process, I don't want to go into details, but they are able to find the path. I'll show you a simulation here to show how a kind of very elementary cell can emerge. And here, this is the membrane emerging. So for the membrane here, you see this is a double layer membrane, which is well known by biologists interested in cell. And I'll show you some of those processes. In fact, neural network and even deep learning neural network, which is becoming so popular these last years, is a form of emergent processes, because none of the neuron here, for instance, know that at the end, they'll be able to self-drive a car, because none of the neuron alone can drive a car, but all the neurons together are able to drive a car. So my definition of emergent computation, which is my vision of it, and I know it has been highly criticized, and there is a lot of discussion going on this notion, is that, well, in order to get emergent computation, you need to have obviously the parts. But what is an ingredient is that you need to have basically 2 observers, a macro observers and a micro observers. So a micro observer is the one that just sees the part, here the single part here. And you have here this macro guy, macro with an A, and this is the micro with an I, but this is the the macro guy. And whenever it observes the system as a whole, so it has this capacity to observe it in space and in time. So by that I mean that it can see the whole things, and this is possible when you have the screen of a computer. You can see the whole things. So when you have this global observation, you see you have to create a second semantic domain. You have to create new words, new expressions in order to describe what you see. And then you have those 2 observers, the micro one and the micro one. The whole things in emergence is trying to understand how you connect back this guy with the micro one, how you do connect the 2 observers. Just to give you an example here, the global observer is, observing the jam here of cars, and obviously the micro observer is yourself driving a car, and you can say that the jam is moving at 1 kilometers per hour, but it's pretty sure that you inside the car, you're trying to make movement in order to escape this jam. And I was just telling you about the ant example and a neural network example. My definition of emergence is that biology has a word to say here. And I guess that the three needed ingredients that biology provides for a collective system to be strongly emergent is the idea that, and think again, about my robots here. Why do those two robots connect? They do connect because they have an observer here, and allow me to call it an observer. You'll see it makes sense. You have an observer here, which is the gap, and the gap is observing the robot to some extent, because the gap is the reason why the robots connect together. If you didn't have the gap, the robot wouldn't connect. So you need the certain, environment which explain why the simple agent, the simple part, decide to connect together. There is an environment that needs to play a lead role here. And the third ingredient is the natural selection. Because if you didn't have natural selection, there would be no reason why those agents connect. They have to connect, otherwise they died. And if they died, so there is some kind of natural selection taking place here. I wrote many papers on that, but I believe that emergent phenomena, according to me, only belong to biology, because in fact, biology provide those ingredients. Just to give you another example, if you're interested in the collective flocking behavior of birds, something that either the biologist or the physicist are a lot interested in, trying to understand how there is a collection of flying birds. A physicist will not so much be interested in answering the question, why are they flying this way? It will just try to capture the parameters of those flying birds and try to map the simulation to the reality. While a biologist will be able to answer the question, why? And for instance, the birds would fly this way because that make them saving some form of energy, because they just help themselves to fly in a more economical way, or perhaps this flocking behavior allows the bird to escape some form of a predation, for instance. But you need to have a kind of theological explanation of why those birds are flying this way. So this was the first part. So I'm trying to split it in equal parts, and don't hesitate. Obviously, during the after that, could use the chat window for asking questions about those many things that I already told and discussed. As I was telling, this notion of randomness, emergence, of replication, they still raise an incredible amount of debates, and no one really agree on that. But I give my own view, and hopefully you might agree and disagree on that. So the second part will be perhaps even more interested to you. And I'm going to try to compile some of the works done in my community, this artificial life community, to show you how the researchers in this community, produce and their own understanding of what is the succession of steps that drives to what we usually call a living organism. And it's interesting to know that they might disagree on where to stop. At what moment do we decide that something is living? Do we need, for instance, natural selection? Do we need evolution, if you want, in order for something to be described as a living process? Or can we stop earlier? You'll see that they might disagree on where to stop and where, at what moment can we decide that this thing is living. So the first step for most of the researchers in I think this, what I'm going to tell you is rather perhaps not unanimous, but yet I would say that most of the thinkers and most of the researchers and scientists working in this community of a life might somewhat agree on my scenario of life, and they might work on different steps of this scenario, but the scenario that I'm going to cover with you, might, I think, make them agreeing on that. So, for instance, the departure starting point would be, you have a bunch of molecules, and you need to have an appearance which is a closed chemical reaction network, and that closed chemical reaction network just means that In fact, this is, excuse me for the French speaking here, because, you know, I don't know why, but I think most of the quoted came from some French biologists. And for instance, there was a biologist in 52 who say, but I'm going to read it in English. Life is an open system able to self-maintain, shaped by organic chemical reactions, catalyzed step after step and at a certain ambient temperature by catalyzers. So you have catalyzers here, catalyzers which are organic, complex, whatever, but those catalyzers are themselves produced by the system. So you have this notion of autocatalysis, where, you know, some of the product of this reaction network are able to catalyze themselves, the reaction taking place in the network. This is the reason why you have some form of, you know, closeness in this reaction network. And in fact, this notion of catalysis is central in this first step of the appearance of life. And you know, the study on closed reaction networks is a very common study in theoretical biology. I'm not you know, telling you anything kind of original and new here. There are a lot of famous metabolic networks, closed reaction networks in theoretical biology and in organic chemistry, like the Krebs cycle. Here, this is a cycle of a glycolysis reaction. And interestingly enough, here you see real molecules, you know, you see real with a shape. And what if you're an artificial life scientist, you're not going to play, most of them are not going to play with real molecules, because you don't need this complexity in order, for instance, to study, according to them. I'm not saying they're right. I'm just saying that they do this dump in abstraction. This is Walter Fontana and this Stuart Kaufman, two very colleague scientists in A Life. You see that they are playing, this is an excerpt of a very famous paper by Fontana, that was interesting to understand how self-maintained autocatalytic networks can emerge in a kind of vessel where you have a lot of potential reactions. And you see that the molecule here, they're just as simple as just letter, A, B, A. So you could imagine, but this is just a reaction that are polymerization reactions. So, you know, you create bigger chain, or, you know, hydrolysis reaction where you cut those chains. So you create big chain and you cut the chains. And then by this kind of two very antagonistic process, you know, assembling and cutting of, then you obviously can create loops. And this is what Fontana was showing, is that the only thing that is able to self-maintain or survive in a very complex reaction networks are in fact those autocatalytic networks here. And it's interesting to know that they just played with letters, and in fact even more, Fontana is famous to have played with lambda calculus, so which is a very strong theoretical you know, matter of computer science. And so he made a simulation where you could you know, send and recruit new molecules in this vessel, this reactor, and then you'll see that those metabolic network, whatever, will just emerge, because they just create loops of different complexity. And once you have a loop, you have a self-maintenance in the networks. In my lab, 20 years ago, I started developing this kind of simulation, but I was not completely satisfied with the level of abstraction of Walter Fontana and Stu Kaufman. I wanted at least to be a little bit closer to reality, the chemical reality, and not to play with just you know, string of letters, but also to play with more realistic molecules, so like graph. So in my case, the molecule would become kind of graph. So you see here some kind of graph, but the idea was basically the same. I have a simulation where you put a lot of those graphs and you have some reaction taking place in between the graphs. So you create new graphs or you destroy existing graphs. And then you have you can, at the end of your simulation, you'll see appearing some kind of chemical reaction networks. So the model I built back in time, so there's papers describing that, was to use a description of molecule called synton, where here you see a graph which is an organic molecule with nitrogen, two hydrogen atoms, a carbon one, and this is another molecules. And those symptoms, they can obviously react among themselves, and the reaction process it's itself described by some kind of symptom. So here you might have a kind of a crossover type of reaction where part of this molecule is being exchanged with part of this molecule here. So you see, you can also code the different type of reaction mechanism, and then playing with that model, you can put a lot of molecules, put a lot of reaction schemes, and see the kind of reaction network that emerges out of it. And the big difference with what my predecessor, like Stu Kaufman and Walter Fontana, were proposing is this fact that we try to keep at least with more realism in the way we shape the molecule. In my community, for instance, there have been people also trying to understand what kind of topology those reactive networks can have. This is an interesting topic also in A life to understand and to analyze the topology of the reaction networks. And you may know that in theoretical biology, but also in physics, the study of the topology of network has become a kind of a very fashionable type of study. You have a scale-free network, you may have heard about that, or random networks. And in fact, it seemed that the topology of a network, for instance, something around the planet, in fact, it was one of the most original pioneering work of James Lovelock back in times, when he was thinking about this Gaia hypothesis, that the network, the reaction networks, above the planet or whatever, might have a certain topology which might have a signature of interesting phenomena like living processes. We did some work, and I remember many discussions I had back in time with Muriel Gargaud, Hervé, and some of my former colleagues. I must admit that I'm not so much interacting with them any longer. And I regret that, because this was a very productive part of my scientific life. We're trying to simulate astrochemical type of chemistry, which is a very original chemistry, a very complex one with very simple molecules, but yet a very exotic one. And we have some idea of the kind of networks, chemical networks, because there's been some database with those reaction taking place in between the stars, so very far from here. And we were able to create a simulator that was able to generate network of of astrochemical reactions. This was a network that was generated. So then we could try to match our simulated networks with real networks that exist in the database, and so that helps you to understand what could be the reaction mechanisms that generate this kind of networks. Another very interesting work that I did with Raphaël Plasson, who was the main author of this work, is once you have a network of chemical network, you may also explain a lot of other interesting things taking place in the biology, in the living process. One of them is the homochirality, and the fact that most of the molecules in life, in biology, seems to have one favorite orientation, while you have a perfect symmetry. You famous, let me show you a slide here and I come back on those things. You have this famous symmetrical figures between the right hand and left hand. So in terms of energy, in terms of a lot of things, those two orientations are exactly the same, but there are still two orientations. Like one is the mirror reflection of the other one. And we wrote many papers trying to understand how you see this title, spontaneous emergence of homochirality in non-catalytic system. And so we played with a very simple reaction network that was inspired by prebiotic biology, prebiotic chemistry here. The reaction we were interested in was mainly three types. So we had here an activation reaction. So for instance, here is the whatever, and here we're just also playing with very simple molecule or whatever simple chain called LD for the orientation, L. This is the typical letter of a homochirality study. So here you need first to activate those molecule here, the L and the D. And then you have here a polymerization. So L star plus L give LL. You may have an hydrolysis. So you have you have those two antagonistic reactions that I was telling before. In fact, you creating bigger chain, and you're reducing the chain, so you enlarge and cut, and this is so you have a polymerization and depolymerization reaction. And the most interesting one, in order to understand homochirology, is a reaction called epimerization, where you see here you have an L which becomes a D. So a L changes its orientation just because it is close, it is connected with a D. So the L will just take the shape of a D, and you have the same thing here. L can become a D. So you have this epimerization reaction here, right? And when you take all those reactions together and you create a reaction networks, and you can do it with my previous chemistry that I was telling before, this Sinton chemistry. So we have replicated, we've done just the mathematics of that, or we've even done a full simulation where you have a real molecule here, well, real molecule graph, and you can do the polymerization here. So we're just, making the chain bigger, the depolymerization, so making the chain smaller, so just cutting the chain. And this whole system is called the APET system, due to the name of the reaction here. And what happened is that you have, in that system, the natural emergent phenomena is a symmetry breaking. So what's going to happen, this is what you see in this graph, is that all the molecules, whatever chain, they will just, flip, or if you want, switch to one orientation. And that is due to the fact that you have an autocatalytic loop. So you have an exponential process taking place. And most of if you're interested in symmetry breaking phenomena in physics, symmetry breaking takes place when you have a self-feedback things. You need to have a positive feedback to have a symmetry breaking. And this is exactly what happened here. You have a positive feedback because you have an autocatalytic process. In fact, you have this loop of, bigger chain and reduced chain, and that creates a kind of autocatalytic loop. And you have a self-amplification of one orientation to the detriment of the other orientation. So, and this raised a lot of, attention when the paper was published, because it seems to be one way. There's been other attempts to explain. I'm not in that case, I was helping my theoretical biologist, who is Raphaël Plasson, to make the simulation. But I must admit that the whole idea came from Raphaël Plasson, who is a very brilliant, theoretical biologist, but still very interesting in using computer simulation to teach and to explain what he's interested in. And here you have obviously a key mechanism is autocatalysis. And while the autocatalysis takes place, in general, you can have, whenever you have a kind of call it chemical self-replication. So here you have this molecule AB, which when it encounters an A and B can give two AB. So you have different type of autocatalysis taking place. You can have a kind of a self-replication chemistry or biological self-replication. Raphaël Plasson still, is a very prolix guy. Wrote another very key paper entitled Autocatalysis, because autocatalysis is still a very fuzzy notion. It's a very important notion, to understand most of the phenomena in theoretical biology. But yes, the definition of it is still the object of debate, of controversies. And Raphaël Plasson, in two papers, tried to clarify the definition of autocatalysis. And for himself, and this is related with most of the simulation he did before, for himself, you have autocatalysis when you have the only possible definition. It is his vision, but if you want, we can discuss and defend it. I can defend it later on during the discussion. But according to him, you have autocatalysis. The definition of autocatalysis needs to be a kinetic one. So you have autocatalysis when you have some kind of exponential processes here. So you see, this is typically the mathematical equation of an exponential increase here. You see this, the velocity is, proportional to the concentration with some power here. So you need to have this kinetic definition of autocatalysis. And this is this vision, autocatalysis is a kinetic phenomenon. And he tried to see what kind of a structure, what kind of a network, what kind of chemistry can give rise to this kinetic signature. But yet, you have catalysis, because sometimes people believe that you need to have some kind of a reaction or networks to get autocatalysis, but not, because some of them might not lead to an exponential increase. Some might lead to an exponential increase, and this exponential increase is definitely, according to him, and I tend to follow what he proposed, the signature of autocatalysis. That is for autocatalysis and reaction networks. I need to go forward, but I think I'll try to keep my time. So once you have autocatalysis, it's not life. For some people, that could just stop there, and say, okay, you have life when you have a self-maintained and even autocatalytic reaction networks, okay? But for another very important thinker, a very important scientist in the theoretical biology, named Francisco Varela, who's been a very key figure to my whole, scientific life. I did a lot of theoretical publication and works with him. Not so much on autopoiesis, but on other things. Francisco Varela proposed back in '74 his own definition of life, and according to him, life was an autopoietic system. And life is a system that, and I just translate that again, sorry for the French. Life is a system that can self-maintain by exploiting, par l'exploitation de la production de ses constituants, by exploiting some energy to produce its constituents. So this is basically the autocatalytic network that you can see here. But that is more important because there is an additional ingredient here, which is this membrane here. The system is circumscribed spatially. So you have a kind of spatial individuation of your system by a membrane, which is semi-permeable, of its composition. So what is really important is that you have a kind of a two self-generated system. The chemistry will generate the component of a membrane, and the membrane itself takes an important role in the chemistry because the membrane help to catalyze the chemistry taking place here. For instance, the membrane could concentrate by providing any escape. The membrane can concentrate the chemical components which are being encapsulated, closed in the membrane, and they can just meet themselves and react. In fact, there was a famous autopoietic software. If you're interested, I could go further to describe it to you, but that was my thing I did, where you have a very simple chemistry here with a catalyzer, which is this green guy. And when there's a reaction taking place, the reaction create the link of the membrane. And you see, so that the membrane can, concentrate the chemical component and the catalyzer here, the green guy. And so the membrane plays an active role here. But the chemistry itself plays an active role because the chemistry is able to self-repair the membrane. So we have two self-maintaining systems, the chemistry and the membrane, which I think is a very nice illustration of autopoiesis. If you're interested in the autopoietic definition of life, I think you should have a look on this very nice cellular automata that is kind of easy to program and kind of easy to understand. So Varela was very interesting in his kind of paradoxical type of ideas. It's often when you read Varela's prose, you might be a little bit lost because there is a lot of paradoxical claims. But the paradoxical claim is the fact that you have two entrenched, self-intertwined systems. One is necessary for the other to exist, and this one is necessary for the first one to exist. So you have a coupling of those two systems, and you'll see that. And the best way to understand this kind of coupling is computer science. It's very hard to understand those kind of things if you don't make it in a computer software. So just for that, software plays a key role. Mathematically, it's really hard to see how the physics of the membrane can help a chemical reaction to take place, and how a chemical reaction can generate the components of the membrane. So just the interplay between those two, kind of a self-maintained and autocatalytic system, you need computer software. You need software to be able to play with that and illustrate that. Membrane, I'll say 2 or 3 things about it. membrane is a very important component of cell, obviously, because that is, they play the role of individuation cell, of, you know, encapsulate the cell. Most of the membrane are, you know, the one that are, you know, interesting theoretical biologists. There was bilayers membrane, which are done by those phospholipid here, which are mainly two parts, one which is being repulsed by water and one which is being attracted by water. And there's been a lot of simulation in my community trying to understand how a membrane can shape. And I'm going to show you, for instance, one very simple simulation. I hope you can see it. And if you don't, let me know. So this is things I've programmed myself, where you're going to see a reactor with water here in the middle of it. And you see that you have you have kind of a phospholipid here with the two heads, the black head and the red heads. And the red head is being repulsed by water. So it's more repulsed by water. And the reason why you see this kind of spontaneous bilayer, you know, taking place. And you see that who create those here lipids are those pink guy. Whenever they meet a gray guy here, they create those B head type of molecules here. And if you see that, if you let running that in time, you're gonna see spontaneously a cell emerging with the, you know, the membrane, you know, closing itself around the chemical compounds that create the membrane. So it's not far away from the vision of Varela, and you can play with that. And again, it's not realist in the sense that this very simple molecules were so simple, but yet, you know, it tells you something. You know, for instance, it can just tell you that it works. You know, it really at the end, the membrane will, you know, close on itself, will encapsulate the chemical compound that are the one responsible for the membrane. So it's another kind of autopoietic systems that you can play it. And once the cell is closed, so once you have really conclude the membrane of a cell, and it's the end. You know, then the cell will stay like that, and it will, you know, it's a kind of a what a physicist would call a spatio-temporal attractor. You know, at the end it will remain like it keep changing in time. You know, in the sense that as you can see, the shape is still being influenced by the water here, the blue guy of the water, but it will remain closed, and you'll see the conclusion of the membrane is here to take place. And whenever you launch with simulation, you will always get the similar kind of result. So we don't need to wait until the end, but I think you get the point of my simulation. Okay, so that was Varela, and then other people have tried to, without any template, without any DNA, try to make those minimal cell just self-replicate. Remember that the chemistry inside can be autocatalytic, so you can have a chemical reaction network that can grow, you know, and then you could imagine that at a certain moment in time, if you have new component in the membrane, so the membrane is growing, the chemical network is growing, then we can reach a certain instability threshold where the cell will split in two. Some people in my community have tried to achieve so, and I could say that, according to me, that would be the definition of life given by Luisi. In fact, Pierluigi Luisi, in his simulation of life, and then he's doing a lot of study with the, you know, with a kind of soap bubble, you know, very simple type of a cell, and he's trying to understand how those cells can shape and can self-reproduce without any need for any DNA, any kind of a template, and just and there's been a lot of simulation, a lot of attempt. Even myself, you remember the cell that I was just showing to you, I've played a lot in trying to have a kind of symmetry breaking here for this elementary cell to be able to split in two and to split apart and to create two cells out of it. It's been very hard, and it's not an easy process, and I I've failed most of the time, but this is kind of a thing you can play with software. And but this is where, according to some other authors, and one of them is Tibor Ganti, that I'm going to present in my next slide. This is where some people say that biology takes over the chemistry, and the key ingredient in order to have self-replication is to add a fundamental component, which is what Tibor Ganti in the coming slide used to call an informative template. This is a Tibor Ganti, who was an Hungarian, very famous Hungarian biochemistry, which in 52, as you know, again, much before, it's interesting because it was again much before the discovery of DNA. But Tibor Ganti, who he called an A-life researcher, invented a theory called the chemoton. And in the chemoton, this is his definition of a chemoton: a living system must constitute a unique individual who must be able of a certain metabolism. So this is the idea of this internal, chemical reaction networks. Must be stable, must possess, and this is where this, informative template takes an important role. So it has to possess here this informative template. So if you want, this chemoton is a mix of 3 autocatalytic networks, or 3 autocatalytic process. So the chemical network here, the membrane here, and a DNA template, here an informational template. And in fact, this is my illustration of a chemoton, but this is his own illustration of a chemoton back to his paper, his 52 paper. And you see that there is some kind of a here, illustration, but he put everything in mathematics, and you can see the 3 system, the template, the membrane, and the chemical reaction networks. And this is a whole, autocatalytic process. So you see that you have 1, which plus some ingredients give you 2. So at the end, it explains, self-replication, because you self-replicate simultaneously the informative template, the chemical reaction network, and the encapsulating membrane. And so this is a chemoton model, which is basically 3 co-sustainable systems. What is really important too is that, a little bit like in Varela's theory, those 3 autocatalytic systems, they are coupled. The membrane needs the chemical reactions, it needs a template. So, and in fact, everything has been put into, this was the original papers. So you see that it was deeply mathematical, a lot of the differential equations. So you had the template subsystem, the metabolic subsystem, and the membrane subsystem. So you have the 3 subsystems which were, catalyzing each other. So you had to share some components in those 3 subsystems. This is a very fascinating, pioneering work. You have to imagine that this was written in the 50s, and a little bit like Varela. These are really the pioneers of the my A-life community. Then once you have the DNA template, where for a lot of authors that would be just enough. You could even stop before the DNA template. But once you have a DNA template, then came another possibility, another functionality of this DNA template is this coding functionality, and the fact that the DNA code for something, and the DNA code for, this is what I illustrated here, for some tools of the cell that are those proteins that can play and that are among the constructor of the cell. So the DNA played a coding role here. And this is where another very famous man, and perhaps other famous computer scientist, came on the stage, like Alan Turing, who believe that everything is into the code. And in fact, when you think even in terms of a code, we speak about DNA coding, but the notion of code owes a lot to computer science, and we have a lot of codes in computer science. We have the ASCII code, even a program is often called a code. Show me the code, open code, whatever. This notion of code is fundamental in computer science, and this is mainly due to fabulous figures like Alan Turing and John von Neumann, and the idea that a code can create something or can code something, and that this code can evolve was the basics of a very popular algorithms in my community called genetic algorithms, which is being inspired by not only this notion of code, but also the idea that the code can evolve by means of a genetic operation, genetic mechanism like recombination, this crossover, the fact that you take two strings and you mix them to create two offsprings, which is a mix of those two kids of your two offsprings, and some mutation here. You have a very simple mutation. One is being, transformed is 0, 0 to 1, whatever. And then remember that Richard Dawkins used the same genetic algorithms to illustrate how very complex, creature, whatever, very complex solution can emerge out of very simple mechanisms in computer science. It's just the fact of mixing things, mutating things, so a lot of random process going on can generate, things like the biomorph and very interesting. There are a lot of computer engineers know that, for instance, use genetic algorithms to optimize the motherboards, for instance, the circuits of your computers themselves. They're being and optimized by things like genetic algorithms. This is what I was just telling you. Rich Dawkins used genetic algorithms for explaining how complex shape or complex, creature can emerge out of those very simple. And I guess this is an excellent difference against creationism and Richard Dawkins' writing. So I'm gonna reach the end. So I still have 2 minutes in order to respect my time. So this is my conclusion slide. So I hope you've been interested in discovered A-Life. I think A-Life is still a lot about the same thing. Although, as I was apologizing at the beginning of my talk, I'm not so much active in A-Life any longer. I've been, attending some of the conference. I think the last one was something like 8 years ago. But I would say that most of the researchers in A-Life, which is a very active community, are still somewhere in the crossroad in between computer science and biology, and try to use software mechanism for explaining living system to help us to understand what's life, to help us to understand the basic mechanism of life. They use computer software like the one I was showing to you, like the autopoietic one or the one creating the membrane. They might also use robots. So for instance, some of the experiment we're doing in my lab with with the swarm robots, is typically the kind of things that could also help to understand, for instance, the swarm behavior of real insect, or the swarm behavior of real birds or whatever. They might accept that in all the things I've shown to you, there's this notion of spatio-temporal attractor, the fact that you have something which seems to remain stable, which is an attractor because the things naturally, spontaneously evolve towards that. And an autopoietic system is a kind of a spatio-temporal attractor. So, for Varela, Thibaut Ganti, and perhaps, people in that same community, they could define life as a very special kind of spatio-temporal attractor. There is some spatial form and there is some dynamical process taking place, and like any kind of attractor. That, where to stop the definition of life? Do you need 2? Do you need 3? Do you need 4 co-generation or co-sustainable system? You remember that Varela wanted to have 2 of them, the chemical reaction network in the membrane and this co-sustain mechanism, while Ganti had a 3rd one, which was the template, the DNA template. And was that enough? Do we need evolution, for instance, to have life, etc., etc.? You see, some people believe that you need evolution for having life. So life begins with evolution, but some of us might just say that life has been necessary for evolution to proceed. And if you didn't have the self-maintained chemical reaction network, the self-encapsulating membrane, etc., then you wouldn't have the basic and the starter ingredients for the evolution to take place. So at least you need those things. And this is where the system, to some extent, graft to each other. You need the chemical reaction network in order to get the membrane. Then once you have a membrane, that you have a DNA template, and etc., etc. And at the end, once you have all those ingredients, evolution can start. But the definition of life, which, as I was telling you, is not at all given by any artificial life researcher. They will not dare to, they will never, pretend to give any definition. And I guess that they try to, show many things, and perhaps at the end, like often in science, and the definition of life might be the object of some kind of a consensual decision or agreement. Okay, I think I'm going to stop here. I guess I've used my time now, so this is my last slide. Thanks for attending.
Okay, so thank you very much, Hugues. So I'm reorganizing my screen.
Do you want me to stop the sharing?
It's up to you. If you can use your slides to.
If it is needed, I will use them. Yeah.
Yes. Okay. So I can see there are already some questions. So a short one from Alexandre. Do you have a YouTube channel?
No, I have a YouTube video, but myself, I'm too old for this kind of thing. I'm a generation who doesn't see the point.
Okay, so Maria is asking, is it true that autocatalysis allows the breaking of chiral symmetry?
Exactly.
What is the interest in doing this? For instance, without the property of chirality, the amount of amino acids to be created is reduced, I think. Thanks for your talk. Very difficult, but very interesting.
So this is where I can reach very soon my limits in this work on homochirality. Let me tell you, is a work that originated in a chemistry group at Montpellier that Hervé knows quite well. Those people from Montpellier, like Robert Pascal, Raphaël Plasson, etc. And they proposed this chemical reaction network called the APED one, which seems to be they seem to be very much interested in that, and there seems to be kind of an interesting pre-metabolic or whatever reaction networks with those 3-4 key reaction schemes. This polymerization, depolymerization, epimerization, and activation. those 4 reaction schemes. And the part I played in these groups was to put everything into a code, into a simulation, together with them, obviously, but we were not so good in coding, so we did it all together, and show that if you have this reaction, spontaneously you have symmetry breaking, so that all the whatever the molecule that will appear in the system will be all L or all D. This is what happened at the end of the system. And so why so? Because in fact, it's a real symmetry breaking. You have normally an equal part of it, but once you have a little delta for whatever reason, you have just a little delta. And in fact, when you launch the simulation, it can be all D or can be all L, depending on it's enough to have a very simple perturbation like any kind of symmetry breaking. And then you have this. So no, I know that there's been a lot of discussion after this paper publication, but if you want to understand more about it, and I would definitely, I can explain you what happened in terms of computer science. About the chemistry, the realism of the chemistry, for me, all those things are L and D, and as just a bunch of differential equations and a bunch of Python code. And but Raphaël Plasson, who's the main author of this paper, is professor at I guess Montpellier, so he's still a very active chemistry, and I think he would be delighted to share with you any question, any comment you'd like. It's a long time I've done this work, so I think Raphael would be the best guy to interact with you about that. But symmetry break, I can explain you the symmetry breaking phenomena and the fact that you have this exponential breaking things, but about the realism of the chemistry underlying those things, I'm not so good.
But to induce this symmetry breaking, you need a little delta at the beginning.
Yeah, you always need some kind of delta.
In the code, you force a small delta.
Yeah, the small delta is being amplified, and this is due to the self-amplifying looping things where you have, as you remember, a big chain, break chain. So you have this kind of a self-amplifier looping, and this explain why. But it's a very complex process. Even if you look at the equations, it's not obvious, and it has surprised a lot of people. But at the end, you have a symmetry breaking phenomena. Yeah, you have a little delta, and everyone converges towards one orientation. Yeah.
Okay, thank you. A question from Guillaume. Nice talk. Thanks a lot. Do you think that it is possible to model a cell creation, for example, with an ab initio simulation using fundamental interaction between atoms and molecules?
Well, this is a nice question again. There's been attempt, and so the idea of trying to create a minimal cell has been it's interesting history of A life, because Pierluigi Luisi, who is, as I was telling, a very famous Italian biologist, together with some computer scientists back in, I think it was in the starting of this millennium, so 20 years ago, they created a lab in Italy at Venice, and they wanted both to do a minimal cell with real ingredients by using the minimal DNA that they could imagine, very simple molecules taking part of a membrane, and a very simple, kind of a reaction chemistry or chemical networks, and they did it with real chemical lab, with real chemists. And beside that, together with them, there were computer scientists trying to simulate everything they did with a very simple, what level of simplicity we had. And it's interesting that they failed. Both of them failed. And I must admit that I haven't, I've stopped to follow what we're doing. But I think if you're interested, you could certainly have see things about them. But they failed to create, I think, even in real chemistry, Luisi, I think he was not so much satisfied by the result obtained. And in fact, this laboratory has to close after some years. With respect to the computer simulation, this is what you were asking me. I don't know to what level of precision you need to go in order to, and it also depends on what you call a minimal cell. To some extent, an autopoiesis, I've shown you 2 simulations of minimal cell. So you might obviously dislike that, saying that it's too simple. But I guess that some of the basic ingredients of why a cell exists are still there. I think that you need to have some kind of internal chemistry. You need to have those molecules that are able to, create this membrane, and by being, repulsed by water. So those things, this is what you read in, classic theoretical biology book. And if you implement that with a certain level, even with a very high level of abstraction in the computers, you find things, you discovered things like, the closing of a membrane. Remember when I show you the simulation, it was not obvious, but it would work. So perhaps the conclusion of that would be that perhaps the living ingredient, the chemical ingredient, in order to get a minimal cell, might not be as complicated as the one that we today have in real chemistry. So that we could abstract a lot out of those complexity in order to get what life is. And this is why even the real biologists or real biochemists, they wanted to be able to produce life with very minimal type of chemistry, very simple molecule, constituting the membrane, or even constituting the chemical reaction network, or even a very simple DNA. So they wanted to reach the most simple thing in order to get a kind of a simulation. In simulation seems to be not so much complicated. For instance, the closeness of a membrane, those things seems to be able to be done in simulation. But would that satisfy a theoretical biologist? This is another point. It's a discussion taking place between computer scientists and biologists.
Okay, thank you, Hugues. So next question is from Ricardo. I'm not sure, Ricardo, you will let us know because you talk about autopoiesis, but I think you asked the question before. Hugues talked about this in the talk, but maybe you have follow-up question. Go ahead.
Could you?
Yes, we can.
Could you listen to me?
Yeah,
I do hear you well. No problem.
Okay. Thank you. Well, my question was initially about that autopoiesis, and it was before you told us about Varela, and so my question was about organization. And since you showed us this example of the robots that they can form, various of them can form this pattern of behavior. And so I wonder how much the properties of the components are defining the way they can organize as a whole, the whole set of them. And so how is organization contained in the properties of the components, or organization is completely an emergent property? That was my question. But I want to take advantage of this opportunity to ask something about autocatalysis and organization, and autopoiesis. I suppose that autocatalysis is a reaction that can drain all the substrates, and so it can lead to equilibrium very rapidly. And so perhaps is it needed the coupling of several autocatalysis to compensate this harmful result of absorbing all the nutrients? Is it necessary to have several autocatalysis coupled together to avoid this completely draining of all the nutrients? That's my second question. Thank you.
Well, I understood your first question. I'm not so sure I understood the second one. So perhaps I can try to answer the first one, and then perhaps you could repeat, and I'll try to answer the second one afterwards.
Yeah, sure.
So for the first one, well, it's obviously you need, for instance, in the case of robots, you need the part to be able to do something in order to create the whole, in the sense that the part needs to have some rules. For instance, in the case of a robot, they just need to be able to connect to each other, so to be able to pinch to each other. And they also have needs way to communicate. If you are interested, you can look at some YouTube video where you see that the robots locally, they do see each other, because otherwise this wouldn't work. So the part has to be able to do something in order at least to have some local interaction with other parts. And this is true of most of the interesting emergent phenomena. Even in cellular automata, if you are familiar with cellular automata, which is a very popular type of software in theoretical biology and theoretical physics too, the part have some simple rules, but those rules make the part to interact among themselves. And then you have those collective phenomena that you're observing that emerges. But obviously not all the parts and not all the rules of the part, not all the behavior of the part would give that kind of interesting things. For instance, in the case of a cellular automata, one of the most interesting emergent is, for instance, the game of life, which is a very popular cellular automata. But this only takes place because the part was satisfying, was executing two very simple rules, but very simple still. But those two rules give the game of life. If you change the rules, you don't get the game of life. So obviously the part has to behave in a certain way for the whole things to emerge. That left totally open this notion of emergent. As I told you, this is in fact, although I was very friend with Francisco Varela, this is definitely, for instance, a point of disagreement between myself and himself, in the sense that I never defended this notion of emergence and things like cellular automata, because Varela wanted, according to Varela, you only had emergence if you had a certain form of downward causation, in the sense that the whole has to influence to some extent the part, otherwise you wouldn't have any emergence. And for me, who is a computer scientist or closer to a physicist, that doesn't make any sense. You know, the whole thing can only be bottom up, but that would be a long discussion to have. Now, for the second point, I can just tell you that this autocatalysis notion is for me was a very confusing one, and because, for instance, there are authors like Stuart Kaufman, Walter Fontana, and even Francisco Varela himself, who didn't need the kinetic explosion for having autocatalysis. For them, a self-maintained network was just enough, was an autocatalytic network. So they were associating autocatalysis with self-maintenance of the network, so a kind of stable attractor, some form of stability. While if you think about what I've shown you later, the works of Raphael Plasson and some of simulation we did, you have autocatalysis should create an exponential growth, you know, a kind of.
Yes.
So this is a big difference in the definition we gave, which is in fact closer to the one given by Ganti in his schematic works. Right? In fact, you have a splitting because you have this amplification process. But you asked me something else about the coupling of autocatalysis, and then I didn't get you.
Yes, it was because of this exponential growth of autocatalysis that it could drain all the nutrients for the reaction, and so it could achieve equilibrium very rapidly. And equilibrium is the state of non-life. And so when autocatalysis couples together with other autocatalysis, I wonder if that is needed to self-regulate that behavior of draining nutrients, and so it can get in a collective autocatalysis can get this stable state, this steady state instead of equilibrium.
Very nice point. I would definitely recommend you to perhaps. Well, it's true that, you know, even in Ganti's work, the chematon, you have a coupling of 3 autocatalytic systems. In fact, the membrane, the chemistry, and I would definitely recommend you to read the literature of Ganti and those authors that might provide you some way of answering. And again, don't hesitate. You can even add me CC if you want to interact with Raphael Plasson, because he's really the one doing a lot of chemical experiments with autocatalysis. So I think he is the best guy to answer those more kind of chemical type of questions. So sorry, I won't be able to give you a satisfactory answer.
No, it's fine. Thank you very much for this nice presentation. Thank you a lot.
You're welcome.
Okay, thank you, Ricardo. So we are about to run out of time, so maybe 1 or 2 questions. So this one remind me some discussion we had in a few years ago, or many years ago. So do you think there will be a point where we will be forced to redefine our conception of what life is due to the growth and complexity of AI technology? That is, will there be a point where we will recognize AI created by humans as life itself? And there are some other questions that are related to this one.
It's a very interesting point. You know, I think that as a computer scientist, I wouldn't be too much shocked by the possibility to replicate life in with whatever silicon of computer means. I guess that to some extent life is open to a more classical scientific definition, even if we still need to find a consensus about what this definition is. But I think that we could reach a definition of life which could integrate software or robots or whatever. That at a certain moment in time, we could accept that the robot is living, or even a software, a demo or software is living. but keep in mind that in artificial intelligence, and part of it is what is usually called the strong artificial intelligence, there are people that want to go further and hope that once we'll be able to create things like consciousness or subjectivity in software and robots. And this is where I'm much more uncomfortable with that. I tend to believe that the big issue with that is that it's pretty hard to have an acceptable and shareable definition of what is consciousness, what is subjectivity, and while I think we can agree once we'll see a robot or that it definitely could be seen as a, because most of the functionalities that we associate with life are present in this robot or in this software, we could agree that at the end we could give the attribute of living to that. What will be a completely different things, and I think that we'll never be able to achieve so, would be to give to a robot or a software the attribute or whatever of the characterization of being conscious, and to have some form of a subjective feeling of this kind of things, and I guess you could have life without that, and and it's better for robots, because I believe that most of the robots, and I think, will never have some subjective, some feeling, and this kind of thing. So definitely there is a limit of what can the computers do, and those limit has to do with, as I was telling you, consciousness, emotion, subjectivity, which is not so much the problem with artificial life and real life. This is my answer, but I wouldn't be, I understand that this could raise a lot of dispute and disagreement.
Okay, and the last one to be answered live. So I try to pick someone who has not asked question yet this week, if I remember. So yes, why not? Gustav, could development of a computer simulation of human immune system eventually eradicate the need for human testing during development of vaccines?
This is obviously a very interesting point too. I've myself worked a lot with a theoretical immunologist, helping them by computer simulations, and we are reaching a very good level of realism in theoretical immunology with computer simulations, to the point that even the concentrations of our, for instance, antibodies or B cells or whatever the components of the immune system that we see in our computer simulations are very close to the concentration that we see in reality. So the fact that we could use simulation of immune systems in order to test whatever drugs, medicine, treatments, vaccine, or what, I think it's reachable. It's really reachable in, I would say, in the perhaps coming 10 years, and we are gaining a lot in computer power. I remember the time where we're not able to have millions of cells in a computer simulation. This has turned to be possible these days, because computer power has grown so much that our computer simulations are becoming very realistic, even in terms of spatiality, where we can even have a 3D simulation of some immune organ, like the lymph node or the bone marrow, which are very key organ in the functioning of the immune system. Even the vessel, the blood vessels, etc. You can have very interesting computer simulations of that. So I would like to answer yes, and perhaps in a horizon of 10 years, we will be able to test a lot of things, drugs, treatments, vaccine on the simulated immune system. Yeah, I think many immunologists would think like me.